Good afternoon, everyone. My name is Angelina, and for the MSAP of 2020, I did research on the analysis of the electrophysiological responses to trigeminal and non-trigeminal odorants in the olfactory epithelium. So any chemical we smell has to be identified somewhere in our body. For us, this would be the nose. The nose is where the journey of smell begins. Whenever we smell something, and let's say for this example, we're smelling ethanol or alcohol, the airborne molecules of the alcohol will enter our nose and attach to nerve receptors, which identify the molecule and therefore allow us to smell it. So let's dig a little deeper. When any molecule enters our nose, it will travel until it reaches the olfactory epithelium, which is circled on the left image in orange. The olfactory epithelium is where the molecules we smell will attach to different nerve receptors. The different nerve receptors are called olfactory sensory neurons, or OSNs, and those are represented in the right picture and are circled by red. OSNs are capable of responding to multiple odors. In the olfactory epithelium, there is also another nerve. This nerve is called the trigeminal nerve and contributes to pain sensation in the olfactory epithelium as well as in the entire face. And the trigeminal nerve is circled in the right picture in blue. So ethanol is also an irritant and activates the trigeminal fibers in the OE, or olfactory epithelium, which triggers a non-receptive response like sneezing, itching, or coughing. Some molecules are capable of activating both of these systems, while others are only able to activate one. So how does this interaction happen between the molecules and the OSNs? Well, in the OSNs, the odorant or irritant will attach to the cilia of the OSNs. Once the molecule attaches to the cilia, a sequence of reactions is activated and generates the transduction current, which constitutes the first draft of the olfactory information. Similar to the OSNs, once a molecule attaches to a trigeminal fiber, it binds to the TRPA1 or TRPV1 channel. Once a molecule attaches to the channel, it will open, allowing the sodium and calcium to enter the neuron. This flow of molecules inside the neuron triggers trigeminal activation. Now that we understand how we smell and the process of sensing odorants, let's talk about why these two systems are being researched. We are aware that trigeminal and olfactory systems are in the same place, and almost every odor is an irritant and every irritant is an odor. So the two systems are activated together. Right now, we are trying to understand is olfaction and chemesthesis interact, or whether these two interact. And chemesthesis refers to the trigeminal system. So does this interaction influence odor perception or not? By understanding more about these two systems and how they interact, we can understand more not only about smell and pain perception, but also about how multimodal sensory integration works. This can be important in understanding different people's perception and smell. In order to further understand the activation of these systems, we want to observe the transduction mechanisms of the systems and observe if there is a difference in their responses when one transduction cascade, the trigeminal, is unexpressed and when it is functioning. In order to do this, we used two different mouse strains, one of which was a wild type, which is shown on the left. This mice strain has a functioning trigeminal transduction system. On the right is our second mice strain, called the TRPA1/V1 knockout. This mice strain does not have a functioning trigeminal transduction system because the mouse is missing its key receptors in its trigeminal pathway. Comparing the responses to odorants in these two different strains will allow us to identify the trigeminal and the olfactory component of the odor response. In order to view the changes in the transduction cascades, we need to utilize electrophysiology. OSNs create electrical signals by by transducing chemical signals to electrical signals. The EOG measures those electrical signals. The responses of the olfactory epithelium's transduction cascade was recorded by placing an electrode on the surface of the olfactory epithelium, which is shown in the central panel. When the odor stimulus reaches the epithelium, as shown in the panel on the right, we recorded the responses to an allyl isocyanate, or also known as AIC. This is also known as mustard oil, and we also used a penethyl alcohol, which is also known as PEA, and is a rose-like scent. AIC is an irritant and is capable of activating both the olfactory and the trigeminal systems, while PEA is a pure odorant that is used and is capable of activating only the olfactory system. Here, I am going to explain the power spectrum analysis. The power spectrum analysis is a way to represent the FFT. The FFT stands for the fast Fourier transform. The FFT allows me to break down the EOG signal into all its components. With this type of analysis, we can bring to the surface hidden features of the response from the EOG that were calculated so that we could analyze their oscillations. This assists in analyzing hidden features of the response since I am able to zoom in and zoom out of the oscillation response at will. This is the results in graphs from the EOG responses. The x-axis is time in seconds, and the y-axis is the amplitude in millivolts. And the red are the recordings from the TRP-A1/V1 knockout mice, while the black lines are the recordings for the wild type. Here are shown the EOG recordings in response to PEA and AIC in wild type and knockouts. For the responses in the graph, the more negative the response, the bigger is the olfactory response. Stimulation with PEA evokes similar olfactory responses in wild type and knockouts. But for AIC, the EOG responses recorded in knockouts, which are in red, are significantly higher in comparison to wild type, suggesting that in the olfactory epithelium, the trigeminal system is activated by AIC. The x-axis is the time in these graphs, and the y-axis is the amplitude in millivolts. We utilize the high-pass filter in order to filter out the slower transduction cascade response. We also evaluated if there are any processes happening which are usually masked by the transduction cascade. High-pass filtered olfactory responses are shown here. These are high-frequency oscillation events in the response to AIC that are not visible in PEA responses. From time periods of 5 to 5.2 seconds, you can see a higher amplitude in the oscillation. These signals are suggesting that other processes are happening with AIC and were completely masked by the transduction current. The x-axis is the frequency in hertz, and the y-axis is the power in millivolts squared divided by the hertz. After the FFT, the frequencies were represented in a power spectrum analysis. From the power spectrum analysis, we can see that AIC evokes oscillations in a frequency band of 20 to 35 hertz, with a peak at 24 hertz frequency, and have the maximum amplitude or power in the wild-type mouse, but are still present in TRP-A1/V1 knockout mice. To conclude the results, the trigeminal response is supposed to be stronger than the olfactory stimulus. Because of the irritant used, irritants tend to have a larger response. Since we've seen a decrease in the AIC wild type and the AIC knockout types, we can confirm that the knockout strain does work and the olfactory epithelium does experience a change from it. The fact that the TRP-A1/V1 response to AIC decreases in its amplitude makes sense, since the TRP-A1/V1 knockout is missing their channels that initiate trigeminal activation. PEA wild type and knockout had around the same amplitudes because PEA only activates the olfactory system. So it makes sense on how there is no change in the amplitude between the knockout and the wild type. PEA and AIC activate different pathways, which is represented by additional oscillations observed in AIC responses. Trigeminal and olfactory systems interact in the olfactory epithelium, which is shown by the reduction of Eog response to AIC and TRPA1/V1 knockouts. Responses to a trigeminal odorant, or AIC, but not to a pure odorant, PEA, presents a high-frequency oscillation from 20 to 25 Hz. This correlation does not disappear in TRPA1/V1 knockout mice, suggesting that it might not have trigeminal origin, but olfactory origin. That concludes my presentation, and I want to thank everyone who donated to the MSAP program, and a big thanks to my mentor, Federica Genovese, and the RISERT Lab for hosting me for the MSAP summer of 2020. I hope you all enjoy your time here and the rest of the presentations today. Thank you.